At the Demoucelle Parkinson charity, we work hard to ensure the development of a cure for Parkinson's disease. What exactly makes the disease so devastating, and which hurdles do we need to overcome if we want to find a way to treat it? Let us give you a short overview of what we know and don't know about Parkinson's disease. First of all, what is Parkinson's disease really? We will tell you which symptoms patients have to deal with and what the underlying brain problems are that cause them. We will go over the cure and treatment options, which unfortunately are only symptomatic. However, more promising treatments are on the horizon, and we'll tell you what the Demoucelle Parkinson charity is doing to speed things up and how you can be a part of it. But let's start at the beginning. What is Parkinson's disease? These are the facts. More than 5 million people worldwide currently suffer from Parkinson's. The disease is most common in the elderly, but can also occur at a younger age during adult life. The higher the age group, the bigger the percentage of people that get Parkinson's. And in those older than 70, up to 1 in 50 persons will develop Parkinson's. The clinical definition of Parkinson's disease, which describes the cardinal symptoms, is mainly based on the motoric symptoms of the disease. One of the easiest symptoms to notice is tremor. Parkinson's patients cannot control their movements, and that is why they shake. Since it's difficult for them to coordinate, they are often also slow at initiating movement, also suffer from rigidity, and have difficulty in keeping their balance. Dyskinesia, which is involuntary movement, is often mistaken for a symptom of Parkinson's disease, but is actually a side effect of the treatment. Next to problems with movement, there are several additional symptoms that patients have to deal with, especially as the disease progresses. Some of the most common ones include constipation, loss of sense of smell, sleeping problems, mood disorders, speech and swallowing difficulties, or dementia. So what causes all of these different symptoms? This brings us to the pathological definition, which describes the changes in the brain of Parkinson's patients. A specific group of neurons controls the coordination of movement. These neurons send out signals to communicate with other brain cells, which subsequently send out the message to our muscles. These neuron signals are propagated via the chemical dopamine, and that's why these neurons are also called dopaminergic neurons. In Parkinson's patients, the dopaminergic neurons slowly die, resulting in less and less dopamine signaling in the brain. Dopaminergic neurons are located in the center of the brain. They contain a pigment which actually makes them visible when you look at a brain section. On the top right, you can see a big dark colored region where the dopaminergic neurons are. This region is often called substantia nigra, which means black substance. Because these neurons degenerate in the brains of Parkinson's patients, the dark region becomes less and less visible, as is shown in the picture on the bottom right. By the time that symptoms emerge and patients are diagnosed, more than half of the dopaminergic neuron population has already died. This means that when we treat or study patients, we're actually looking at a disease process which is already in its final stages. The loss of dopaminergic neurons causes the movement problems, which are the most typical clinical symptoms. However, there is another peculiar change in the brains of Parkinson's patients. When you look at the brain cells under a microscope, you see that a lot of neurons contain strange spots called Lewy bodies. In the picture, you can see 2 neurons. In the right one, a circle is drawn around the Lewy body. Lewy bodies are named after Frederick Lewy, who was first to describe them in 1912. It's more than 100 years ago. We know now that these Lewy bodies are composed of a large amount of misfolded proteins, but we don't understand very well why these aggregates are formed and how they affect brain function. So now that we know that loss of dopaminergic neurons causes movement problems, why do these neurons die in the first place? This question has puzzled scientists for decades, and while we don't have a definite answer, there are lots of clues. What do patients with Parkinson's disease usually have in common? There are several environmental factors that are associated with Parkinson's disease. First of all, the older you are, the more likely it is you will get the disease. There are also more males than females with Parkinson's, and quite surprisingly, it seems to affect non-smokers more often than smokers. People who drink a lot of caffeine also have a smaller likelihood of getting the disease. There are other factors which may be involved, but so far did not yield conclusive evidence. High exposure to pesticides and certain toxins, for example, have been linked to Parkinson's disease, causing people who live in rural areas or who work as farmers or painters to be more vulnerable to the disease. Low urate levels and limited use of anti-inflammatory drugs seems to be accompanied with a decreased risk for Parkinson's disease as well. Getting regular exercise is good for you, also in terms of Parkinson's. And while suffering from head injury or brain trauma may increase your chances of getting Parkinson's disease. This may be why a relative high number of boxers get the disease later in life. It's important to note that just because these factors are linked to an increased risk, it does not necessarily mean that they actually cause Parkinson's. Interestingly, there are genetic causes for Parkinson's disease. Patients and family often ask whether Parkinson's can be inherited. For a long time, scientists did not think that Parkinson's was genetic, but in rare cases, there were families in which many members were affected. Over the last 15 years, several genes have been identified in such families that indeed cause a heritable form of the disease, also referred to as familial Parkinson's disease. In these cases, disease onset is often at a much younger age. However, familial Parkinson's disease is very, very rare. Only 1 to 5% of all Parkinson's cases are inherited. Nevertheless, the study of these families and the genes that are faulty is quite important, also for patients who suffer from non-familial or so-called sporadic Parkinson's disease. Because everyone suffers from the same disease, with the same symptoms and the same pathological hallmarks, this must mean that the underlying problems are similar. So even though familial Parkinson's cases are rare, we know where in the DNA they have a mistake, and if we can understand what problems these faulty genes cause, this brings us to the general mechanisms underlying the disease, which are likely to be shared by sporadic cases as well. Genetic changes that cause familial PD increase the disease risk massively, but they are very rare. Some examples include the genes LRRK2, Parkin, SNCA, and PINK1. Genetic studies are now focusing on genetic abnormalities that may be more common and do not necessarily cause disease, but could increase the risk of getting Parkinson's. These are called genetic risk factors. So if these genes may lead us to the common disease mechanisms, what have the familial cases taught us so far? Scientists currently focus on 3 main defects in the brain. The first is protein accumulation, which can be toxic to cells. One of the genes that are faulty in familial Parkinson's is SNCA, which codes for the protein alpha-synuclein. This protein happens to be the one that is accumulated in the Lewy bodies found in patients' brains. Clearly, when the misfolding of this protein and its accumulation are involved during some stage in this process. A second recurring problem is the dysfunction of mitochondria. Several genes, including PINK1, Parkin and DJ1, cause problems in the function of mitochondria, which are the power plants of the cell. Thirdly, the death of the dopaminergic neurons could be caused because of transmitting problems in the synapse. Another faulty gene in familial Parkinson's disease, called LRRK2, has been shown to interfere with the proper functioning of signal transmission. The first problem, aggregation of misfolded alpha-synuclein, has been studied most extensively because of the link that's clear between the rare genetic forms and the presence of Lewy bodies in all cases of Parkinson's disease. Currently, scientists hypothesize that when the normal protein gets misfolded, it starts to stick together. These small clumps, called oligomers, further group together in fibrils, forming the Lewy bodies that can be observed in the brain. Interestingly, the spread of Lewy bodies occurs in well-defined stages, as you can see in the picture on the bottom. Lewy bodies are first found centrally in the brain, but as the disease progresses, they spread to more and more brain areas. The problem of protein accumulation is especially relevant since similar processes take place in other neurodegenerative conditions. The brains of Alzheimer's patients, for example, are also filled with aggregates, but in that case they do not consist of alpha-synuclein, but of amyloid beta and tau. The same scenario occurs in Huntington's disease, where a faulty protein called huntin forms aggregates. TDP43 is another such protein which is found in the brains of patients with ALS. There are even a number of diseases, called prion diseases, which have a protein misfolding process that can be transmitted. An example of such a disease is mad cow disease. So all of this evidence points out that somehow alpha-synuclein and the formation of Lewy bodies plays an important role in the disease. Therefore, there's an intense research effort to find a way to block alpha-synuclein aggregate formation. The Demoucelle Parkinson Charity's efforts in the Aerts project are an example of this approach. However, there are few important questions that remain unanswered. Why is the alpha-synuclein protein that misfolds in Parkinson's disease, and not any other protein? Which form is toxic, and how is it toxic? There are also a very small number of Parkinson's cases in which no Lewy bodies were found in the brain. So were these cases even really Parkinson's? And importantly, if alpha-synuclein does not get misfolded, what does the protein normally do in the brain? This is an important question because if we want to block alpha-synuclein in the brain, we don't want to cause other major side effects. A second recurring theme is the role of mitochondrial dysfunction in Parkinson's disease. Mitochondria are the power plants of our cells. They have an interesting structure with two membranes. The inner one has many folds and is required to produce energy. Neurons especially need a lot of energy to transmit signals. So if the efficiency of the power plants is decreased even only by a little bit, this can have major consequences for neuronal health. Some of the faulty genes in familial Parkinson's disease are involved in energy production and the quality control process of the mitochondria. The cells of patients with sporadic Parkinson's disease also display decreased levels of mitochondrial activity. And it's believed that if we can boost mitochondrial activity and energy production, this can also improve the health of the remaining dopaminergic neurons in Parkinson's disease. So thirdly, there's a problem in the synaptic signaling. The synapse is the contact point between one neuron and another, and it is there where they exchange chemical signals to transmit messages. Some of the faulty genes in familial cases of Parkinson's cause problems in the release of chemical signals. And if there's less activity, this may in turn lead to neuronal degeneration. So this brings us back to the main question. What causes Parkinson's disease? Well, although we have good indications, we're not quite sure yet. Most likely, the three processes we just discussed all play a role in the development of disease. It could be a multi-hit process, where small defects in different pathways all contribute to neuronal death. None of these processes explains why the neuronal loss shows such a high degree of specificity for dopaminergic neurons. Another problem is that none of the animal models used to study the role of the faulty genes for familial Parkinson's disease actually develop clear movement symptoms that are reminiscent of Parkinson's disease. So most likely, there's a combinatorial effect of genetic factors and environmental factors, even in familial Parkinson's disease, that eventually causes the disease onset. So now that we understand a bit more about what Parkinson's is, which processes go wrong in the brain, let's look at what can and can't be done to treat it. Treatments for Parkinson's can be divided into three different types. The first is symptomatic treatment. This type of treatment is aimed at compensating for the loss of dopaminergic neurons by making sure the brain has enough dopamine despite the neuronal loss. These types of treatment have no impact on the underlying problem, and the progressive degeneration of neurons is not halted or slowed down. A second type of treatment is neuroprotection. This type of treatment is aimed at protecting the remaining dopaminergic neurons and preventing them from dying further. This will stop the disease getting worse, but it will not cure it. The third and best option would be a restorative treatment, by which we can regenerate the population of dopaminergic cells so that dopamine signaling can be restored back to normal. Unfortunately, the current treatment for Parkinson's is only symptomatic. Levodopa, or L-Dopa, is still the benchmark treatment for Parkinson's disease. In combination with other dopamine metabolism regulators, it helps to replenish the dopamine in the brain. Let's have a look at this diagram. On the right you can see a synapse. The blue end is a dopaminergic neuron, which transmits signals to the green neuron via the release of dopamine. We can't supply dopamine itself to patients because it cannot cross the blood-brain barrier. On the left, in the pink part of the diagram, you see the blood, which is separated from the brain cells in blue. However, dopamine is made from L-Dopa by an enzyme called DCC. L-Dopa can cross the blood-brain barrier, which is why it's used as a Parkinson's treatment. Once in the brain, it can be converted to dopamine. Besides L-Dopa, there are other ways to make sure that there is enough dopamine in the brain. Normally, dopamine will be broken down by MAO-A and B. If these enzymes are inhibited, for example by ladostogel, then the low levels of dopamine will have a better chance at doing their job before they're broken down. In order to make sure that the L-DOPA patients take is not broken down before it reaches the brain, patients can be treated with other inhibitors, such as for example carbidopa. This type of inhibitor actually blocks the conversion of L-DOPA to dopamine, but since it cannot cross the blood-brain barrier, it will not prevent dopamine production in the brain. It will only make sure that the supplied L-DOPA is not converted to dopamine anywhere else where it's not needed, but can all be transported to the brain. L-DOPA, or L-dopamine receptor agonists, and MAO-B inhibitors are approved by the FDA for use in early and advanced Parkinson's disease. COMT inhibitors and apomorphine are approved only for fluctuating PD symptoms. The side effects caused by long-term chronic use are motor fluctuations. Too little dopamine causes lack of movement, too much causes uncontrolled movement. And Parkinson's patients fluctuate between the two states by using L-DOPA. Currently, there's a lot of effort to improve the delivery of dopamine and dopamine metabolism regulators, for example via skin patches, inhalations, or subcutaneous delivery. Besides the pharmacological treatments, there are surgical methods to improve Parkinson's symptoms. Deep brain stimulation is aimed at changing the brain's signaling circuits to compensate for the loss of dopaminergic neurons. While this technique has been very successful for some patients, for other patients this was not the case. Some of them suffer from adverse effects. Current research is aimed at finding which patients would benefit most from which type of procedure, as there are different anatomical regions that can be targeted by deep brain stimulation. Obviously, there are a number of technical challenges involved with brain surgery. In addition to the current symptomatic treatments, there's a lot of ongoing research on neuroprotective molecules. These are all compounds that can delay or perhaps even prevent further degeneration of neurons. You can see a few examples listed here. Unfortunately, they are all still undergoing further testing. The major challenges are that such a compound must not only be protective, it must also cross the blood-brain barrier to reach the dopaminergic neurons. Some compounds that have shown spectacular results in animal models were not successful in patients. One reason for this discrepancy could be that the effects are too small to make a meaningful difference in advanced disease. Remember that by the time of diagnosis, up to 3/4 of all dopaminergic neurons is already dead. So is there no way to regenerate these neurons? This would truly represent a cure for Parkinson's disease. Unfortunately, the answer is no. There are a number of techniques that are currently studied and could one day be used to regenerate neurons. One of them is gene therapy. Using viral vectors, genes that are required to make dopamine be introduced in the right cells, and they would become capable of dopamine production. Another avenue is cell therapy. The idea here would be to supply Parkinson's patients with a new set of dopaminergic neurons, much like a heart transplant or a kidney transplant. Such neurons could be generated from stem cells. Stem cells are cells that have yet to develop and which can be stimulated to form dopaminergic neurons, which can then be implanted. The basics of this research were done using human embryonic stem cells, which has been ethically controversial. However, new methods have been developed by which normal cells can be reprogrammed to form stem cells. The idea would be to take skin cells from patients and reprogram them in the laboratory to convert them to dopaminergic stem cells. All of our cells have the same genetic material, so they all have the potential to develop into any cell in the body, including dopaminergic neurons. Unfortunately, the processes of programming and reprogramming cell fate are only beginning to be unraveled, and it might still take years before we know whether this approach could truly be used to cure Parkinson's. So knowing about Parkinson's disease and its limited treatment, what is the Demoucelle Parkinson charity doing to make a difference in the search for a cure? Let's summarize the current challenges in the field. We know that a large majority of Parkinson's cases is likely caused by multiple factors, including genetic and environmental risk factors. This would also explain the heterogeneous picture of the disease, since the contribution of different symptoms, the age of onset, and the speed of progression are highly variable across patients. We need to better understand the differences between these patient groups, as some treatment approaches might be more useful for some patients than for others. This is already clear, for example, for deep brain stimulation. There is enormous improvement in the delivery of dopamine-related treatments, but the symptomatic treatment will only take us so far. We need to focus on disease-modifying approaches, and this is exactly what we aim to do at the Demoucelle Parkinson charity. Several protective treatments are already in clinical trial, and we have been supporting such trials. While neuroprotection might not always be a cure, it can halt a disease which buys us more time. Importantly, some of these compounds have been previously used in other diseases, and so if they indeed show to be successful for Parkinson's, they can be approved as a treatment much quicker. Restorative treatments are the ultimate goal, but the majority of these approaches still require a lot more work. Nonetheless, insight from genetic studies are starting to bring new targets into clinical studies, including alpha-synuclein blockers and mitochondrial stimulators. Because of the progressive nature of the disease, the earlier we can treat, the better. That's why we need reliable biomarkers to detect the disease in its early stages. Good biomarkers will also allow us to study the effects of potential treatments much better. The Demoucelle Parkinson Charity also actively supports research into biomarker development. By gathering funds, we can help to move the most promising research findings forward. And as an independent organization, we never have to compromise on our goal, which is to speed up a cure for the millions of patients worldwide. You can help us by supporting us financially and by spreading the word about our work. It's not a question of if, but when we will beat Parkinson's disease, and we will not let it slow us down. We take this quite literally, for example during the 20 kilometers of Brussels. It can be done.